Good morning, everyone. We are the group 3 and we are here to discuss about gene therapy. Table of contents. First, introduction. Second, types of gene therapy. Third, approaches in gene therapy. Fourth, vectors in gene therapy. Introduction. What is gene therapy? Gene therapy is the introduction of genes into existing cells to prevent or cure a wide range of diseases. It is a technique for correcting defective genes responsible for disease development. But before we dig deeper to what is gene therapy, let us all know what is gene. A gene is a basic physical and functional unit of heredity. Genes are made up of DNA and each chromosome contains many genes. A gene is a segment of DNA that contains instructions for building one or more molecules that help the body work.
So, what exactly is gene therapy? First, let's start with the basics. This is a gene. Genes are made up of DNA, which are the blueprints to build the enzymes or proteins that do the work for your body. As far as we know, humans have between 20,000 and 25,000 genes. And most people have two copies of each gene inherited from each of our parents. They are the building blocks of our physical makeup, from the color of our hair, to our athletic abilities, to our immune system. But sometimes genes aren't built correctly. And a small adjustment to the DNA within our genes can change how our proteins work, which can affect how we breathe, walk, or digest food properly. To better understand how genes change, here's Dr. Michelle Kalos.
The two types of gene therapy is the somatic cell gene therapy and the germline gene therapy. The somatic cell gene therapy is a therapeutic genes transferred into the somatic cells. Somatic cells are the cells in the body other than sperm and egg cells, which are called germ cells. Like for example, introduction of genes into the bone marrow cells, blood cells, skin cells, and etc. And it will not be inherited later generations. At present, all researchers directed to correct genetic defects in somatic cells. The germline gene therapy is a therapeutic genes transferred into the germ cells. These are the sperm and the egg cells. Like for example, genes introduced into eggs and sperms. It is heritable and can be passed on to later generations. For safety, ethical, and technical reasons, it is not being attempted at present.
Approaches in gene therapy. Number 1. Ex vivo gene therapy. Here are the following ways of ex vivo gene therapy. First is isolating cells with genetic defect from a patient. Then grow the cells in culture. After that, introduce the therapeutic genes, then select genetically corrected cells and grow. And lastly, transplant the modified cells to the patient. Examples: the first gene therapy to correct deficiency of enzyme adenosine deaminase. It is performed on a 4-year-old girl, Ashanti De Silva, was suffering from SCID, severe combined immunodeficiency, caused due to defect in gene coding of ADA. Deoxyadenosine accumulate and destroys T lymphocytes, disrupts immunity, suffer from infectious diseases, and die at a very young age. Ex vivo gene therapy is performed with the genetic alterations of patients' target cells happening outside of the body. In a culture, target cells from the patient are infected with a recombinant virus containing the desired therapeutic gene. These modified cells are then reintroduced into the patient's body, where they produce the needed proteins that correspond to the inserted gene. Number 1, copies of therapeutic gene then gene inserted into the viral DNA. Number 2, target cells removed from patient, then cells grown in culture. Cultured cells are infected with genetically altered virus. Then number 4, cells are reintroduced into body. Then number 5, inside the body, the genetically altered cells produce the desired proteins encoded by the therapeutic DNA. Patients' sample target cells are now genetically altered with therapeutic gene.
So in vivo gene therapy, it refers to direct delivery of therapeutic gene into target cell into patient's body. And also it carried out by viral or non-viral vector systems, and it can be only possible option in patients where individual cells cannot be cultured in vitro in sufficient numbers. And lastly, in vivo gene transfer is necessary when cultured cells cannot be reimplanted in patients effectively. So like for example, in patients with cystic fibrosis, a protein called cystic fibrosis transmembrane regulator, or what we call CFTR, is absent due to a gene defect. So in the absence of CFTR, chloride ions concentrate within the cells and it draws water from surroundings. And this leads to the accumulation of sticky mucus in respiratory tract and lungs, and it treated by in vivo replacement of defective gene by adenovirus vector. So in this picture, first we're gonna do is remove bone marrow stem cells. Then second, use retroviruses to bring the normal gene into the bone marrow stem cells. And the third one is viral recombinant DNA carries normal gene into genome. And lastly, the fourth one is return genetically engineered cells to patient.
Vectors in gene therapy. Vectors in gene therapy to transfer the desired gene into a target cell. A carrier is required. Such vehicles of gene delivery are known as vectors. There are two main classes. First, we have viral sectors, and the second one, we have non-viral vectors. First, let's talk about retrovirus vector system. The recombinant retroviruses have the ability to integrate into the host genome in a stable fashion. It can carry a DNA of size less than 3.4 kb. Replication defective virus particles, target cells dividing, and the adenovirus vector system. Adenovirus within a genome, God vectors, target non-dividing human cell, and common cold adenovirus.
Non-viral vectors. First, we have pure DNA construct. It's direct introduction of pure DNA construct into target tissue. Efficiency of DNA uptake by cells and expressions rather low. Consequently, large quantities of DNA have to be injected periodically. Second, we have DNA molecular conjugates. Commonly used synthetic conjugates is poly-L-lysine bound to specific target cell receptor. Therapeutic DNA is then made to combine with a conjugate to form a complex. It avoids lysosomal breakdown or DNA.
Disadvantages of gene therapy. Long-lasting therapy is not achieved by gene therapy. Due to rapid dividing of cells, benefits of gene therapy is short-lived. Second, immune responses to the transferred gene stimulates a potential rest to gene therapy. Third, viruses used as vectors for gene transfer may cause toxicity, immune responses, and inflammatory reactions in the host. Lastly, disorders caused by defects in multiple genes cannot be treated effectively using gene therapy. Advantages. Gene therapy has the potential to eliminate and prevent hereditary diseases such as cystic fibrosis, AIDSID, and etc. Second, it is possible cure for heart disease, AIDS, and cancer. Third one, it gives someone born with a genetic disease a chance to life. Lastly, it can be used to eradicate diseases from the future generations.
I'm here to conclude the report or our report. So theoretically, gene therapy is the permanent solution for genetic diseases. Some of the genetic diseases that can be cured by this therapy is cancer, but it has several complexities as its current stage. It's not accessible to most people due to its huge cost, especially to those who don't have enough money for the therapy because they use equipments that are expensive, and so it's a huge cost. So a breakthrough may come anytime, and a day may come when almost every diseases will have a gene therapy. So in the near future, if there's some diseases, so mag-need gene therapy and most people cannot afford it. So the last one is gene therapy have the potential to revolutionize the practice of medicine. So here's the quote, a picture is worth 1,000 words. So we must not judge the people of what we see outside without knowing their true personality on the inside. So thank you for listening.